Pharmapal.
Moringa Oleifera or miracle tree pharmacokinetic properties. Hello everyone! Welcome back to our channel. Today, we're diving into some fascinating research about Moringa Oleifera, often referred to as the drumstick tree. This plant has garnered attention for its impressive health benefits, and recent studies have shed light on its antioxidant, antibacterial, and pharmacokinetic properties. So, let's get started. What is Moringa Oleifera? Moringa Oleifera is a tree native to parts of Africa and Asia. It's known for its nutritional value and medicinal properties. The leaves, seeds, and pods are packed with vitamins, minerals, and antioxidants, making them a popular choice in traditional medicine. Key findings from the research. Antioxidant activity. The study demonstrated that Moringa Oleifera leaves exhibit significant antioxidant activity. This was measured using two methods. DPPH scavenging activity. This method assesses the ability of the extract to neutralize free radicals. The results showed that Moringa can effectively reduce oxidative stress. ABTS radical scavenging activity. Similar to DPPH, this method evaluates how well the extract can combat another type of radical. Again, Moringa performed remarkably well. Antibacterial properties. Moringa Oleifera was tested against various bacteria, and the findings were promising. The extract showed strong antibacterial effects, particularly against resistant strains. This suggests that Moringa could be a natural alternative for combating infections. Pharmacokinetic profile of quercetin. One of the key compounds in Moringa is quercetin, which has been studied for its potential as a therapeutic agent. The research utilized the ADMET Lab 2.0 program to assess quercetins. Absorption, how well it is taken up in the body. Distribution, how it spreads throughout the body. Metabolism and excretion, how it is processed and eliminated. The results indicated that quercetin possesses favorable properties for drug development, but it also raised some safety concerns regarding its interaction with certain enzymes in the liver. Understanding key terms. To help you remember the important concepts, here are some key terms explained. Antioxidants, compounds that prevent cell damage by neutralizing free radicals. Antibacterial, substances that kill or inhibit the growth of bacteria. Pharmacokinetics, ADMET, a field that studies how drugs are absorbed, distributed, metabolized, and excreted in the body. Key figures and tables. Figure 1, antioxidant activity of Moringa Oleifera extract. In this figure, we see the results of the DPPH, diphenyl 1-picrohydrazole, radical scavenging assay. This method measures the antioxidant capacity of the extract. What does it show? The higher the scavenging percentage, the more effective the extract is at neutralizing free radicals. Key terminology. DPPH, a stable free radical used to evaluate antioxidant activity. Scavenging activity, the ability of a substance to remove free radicals. Table 1, total phenolic and flavonoid contents. This table summarizes the total phenolic and flavonoid contents of the Moringa Oleifera extract. Why is this important? Both phenolics and flavonoids are known for their antioxidant properties. Key insights. The total phenolic content is expressed as gallic acid equivalents, while flavonoids are measured as epicatechin equivalents. Figure 2, cytotoxicity assay results. Here, we have the results from the MTT assay, which assesses cell viability after treatment with different concentrations of the extract. What to note, the graph illustrates how cell viability decreases with increasing concentrations of the extract. Terminology explained. MTT assay, a colorimetric assay for assessing cell metabolic activity. Cell viability, the proportion of live cells after treatment. Table 2, antibacterial activity. This table lists the antibacterial effects of Moringa Oleifera against various bacterial strains, including their minimum inhibitory concentrations, MIC. Key takeaways. The extract demonstrated significant antibacterial activity, making it a potential candidate for natural antimicrobial agents. Understanding MIC, the lowest concentration of an antimicrobial that will inhibit the visible growth of a microorganism. Pharmacokinetic analysis of quercetin. Figure 3, ADMET profile of quercetin. This radar chart displays the pharmacokinetic properties of quercetin, a major compound in Moringa oleifera. What does it indicate? Quercetin meets several drug-likeness criteria, suggesting its potential as a therapeutic agent. Key terms: ADMET, absorption, distribution, metabolism, excretion, and toxicity. Lipinski's Rule of 5, a set of rules to evaluate the drug-likeness of a compound. Table 3, biological activities of compounds. This table details the biological activities associated with various compounds found in the extract. Highlights: each compound's activity is linked to its chemical structure, showcasing the diverse health benefits of Moringa oleifera. Challenging questions for discussion. As we explore this research, let's consider some challenging questions. In light of your research findings, let's delve into the challenging questions regarding quercetin and Moringa oleifera. 1. What are the implications of quercetin's poor plasma protein binding, ppb, for its therapeutic use? Implications of quercetin's poor plasma protein binding, ppb. Poor plasma protein binding, ppb, can significantly affect the therapeutic use of quercetin. Bioavailability, low ppb suggests that quercetin is less likely to bind to plasma proteins, which may lead to higher free, active, concentrations in the bloodstream, potentially enhancing its therapeutic effects. Toxicity risks, however, this also raises concerns about toxicity. Since quercetin does not bind well to plasma proteins, it may lead to unpredictable pharmacokinetics and increased risk of adverse effects. Dosing considerations: the variability in absorption and distribution necessitates careful dosing strategies to avoid toxicity while maximizing therapeutic benefits. How do the pharmacokinetic characteristics of quercetin influence its potential as a drug candidate? Pharmacokinetic characteristics of quercetin. The pharmacokinetic properties of quercetin influence its potential as a drug candidate in several ways. Absorption and distribution: quercetin shows good absorption in the human intestine and a significant volume of distribution, VD, indicating its ability to reach various tissues effectively. Clearance rate: with a moderate clearance rate, quercetin's duration of action may be shorter, requiring more frequent dosing or the development of sustained-release formulations. Interaction with cytochrome P450: quercetin's interaction with CYP450 enzymes can lead to drug-drug interactions, affecting the metabolism of co-administered drugs, which must be considered in therapeutic applications. 3. What safety concerns arise from quercetin's interaction with cytochrome P450 enzymes? Safety concerns with cytochrome P450 interactions. Quercetin's interactions with cytochrome P450 enzymes present several safety concerns. Drug interactions: inhibition of CYP450 enzymes can lead to increased plasma levels of drugs that are metabolized by these enzymes, raising the risk of toxicity. Metabolic variability: variability in individual metabolism can result in different therapeutic outcomes, making it crucial to monitor patients closely when quercetin is used alongside other medications. Need for toxicology testing: given the potential for adverse effects, further toxicological studies are warranted to ensure safety in human use. 4. In what ways can the findings about Moringa oleifera's antibacterial properties inform future research in natural product pharmacology? Moringa oleifera's antibacterial properties and future research. The findings on Moringa oleifera's antibacterial properties can inform future research in natural product pharmacology in several ways. Natural antibiotic development: the significant antibacterial activity against various pathogens suggests that Moringa extracts could be developed into natural antibiotics, particularly in the face of rising antibiotic resistance. Phytochemical exploration: understanding the specific compounds responsible for antibacterial effects, e.g., quercetin, phenolic acids, can guide the isolation and optimization of these bioactive components for therapeutic use. Combination therapies: future studies could explore the synergistic effects of Moringa extracts with conventional antibiotics to enhance efficacy and reduce resistance development. Let's summarize the key findings of the study. Quercetin, a major component of Moringa oleifera, shows promising pharmacokinetic properties, passing several drug similarity tests. Despite its potential, the study highlights significant safety concerns, particularly regarding its interaction with cytochrome P450 enzymes, which could lead to harmful drug interactions. The extract demonstrates strong antibacterial activity with MIC values ranging from 14 to 26 g/ml, indicating its potential as a natural antimicrobial agent. In conclusion, this study emphasizes the dual nature of quercetin as both a promising therapeutic agent and a compound requiring further toxicological investigation. Future research should focus on evaluating the clinical safety of quercetin formulations, exploring the synergistic effects of Moringa Oleifera extracts with other antimicrobial agents, investigating the broader pharmacological applications of its bioactive compounds. Thank you for joining us in this insightful discussion. Please share your thoughts on the questions posed and continue exploring the world of natural products in pharmacology. Don't forget to like, subscribe, and hit the notification bell for more Journal Club discussions.
Pharmapal.